Hi everyone, welcome to our monthly seminar at Single Cell Spatial Analysis Program. And it's my honor to introduce Dr. Jean Fun. She's assistant professor at Johns Hopkins University in both Department of Biomedical Engineering as well as Computer Science. Before joining GHU, she did her postdoc at Harvard University under Dr. Shelley Zhang's group, and she was involved. She was in the Murfish technology development team and has a lot of analysis contributions on Murfish. Before that, her PhD was also at Harvard University, supervised by Dr. Peter Kurchenko, where she did a lot of work on single-cell methodology development, integrative analysis of genomics, transcriptomics, epigenetics. It's like a really pioneer work at that time. Since 2020, where she joined GHU and start her own lab, she has generated so many fruitful research, over 10 corresponding author publications and many more in preparation. She developed a series of tools for analyzing spatial transcriptomics data from various aspects, including deep convolution, visualization, cell-cell interaction, and so on. And her outstanding research was also recognized by many awards, including the NSF Presidential Early Career Award for Scientists and Engineers, the PACASE Award in 2025, and also Forbes 30 under 30 healthcare, etc. So Dr. Flint's really a pioneer in single-cell spatial field. With that, I would like to leave the time to her to talk about comparative spatial transcriptomics analysis, identifies evidence of off-target protein binding, and this is a long title, 10x genomics in V1, yeah, something, yeah.
Great, perfect. Great, I think I have. Okay, test. Y'all hear me okay? Awesome, great. Great, well, very excited to be here. And yeah, I'll waste no further time to dive into things, as this is a very long talk that I'm gonna try to squeeze a lot of stories into. And yeah, so my lab, we call ourselves the JeffWorks Lab. And these are my students who've led the work that you're about to see. And in general, when we look at a complex tissue, like, say, the mammalian brain, we're very interested in characterizing how different cell types and cell states are organized in this tissue, but also perhaps how they're different in the context of health and disease, and in particular, how the spatial organization can potentially play a role in disease pathogenesis and prognosis. So luckily for us, the spatial resolved transcriptomics field has really blossomed in the last 5 or so years, and has really provided us with the opportunity to begin evaluating these questions. These technologies do span many different what we kind of consider as flavors, either by imaging or by capture and sequencing. And these technologies do differ in terms of not only their method for profiling the capturing and characterizing the transcripts, but also in their, there's a lot of variation in their throughput and their resolution. But very broadly speaking, what these technologies allow us to do is not only see where these cells are in the tissue. This is a coronal section of the mouse brain. Each point is a single cell. But we can also now quantify what are the counts and measures of different genes, in this case, red being high expression, blue being low expression, and appreciate how this excitatory neuron gene has a particular pattern that's very distinct from an inhibitory neuron gene, and likewise and so forth for hundreds to thousands of genes. And of course, since this is a single cell, group, I think you can appreciate how we're able to perform the same kind of dimensionality reduction clustering to identify transcriptionally distinct cell types. But of course, now we can project them back into their original spatial coordinate and appreciate how these cell types are organized in space. So again, many different technologies, many of which have now commercial entities and platforms, making them more accessible. But of course, all of these tools or technologies, they make these measurements, but we really need computational analysis to actually make sense of all this big data and take advantage of the spatial information. So that's what my lab has been focusing on. We develop statistical approaches and machine learning methods as open source computational software for analyzing these spatially resolved omics data sets. And in general, we do think it's very important to make our computational methods easy to use. So we have lots of tutorials for doing lots of different things, but I do encourage you to check them out. And of course, we are also, at the end of the day, we develop these tools, but we're very excited to collaborate with people, including Toshihara at UMich, to then apply these tools and technologies to address interesting biological problems. In this case, we collaborated to apply one spatial transcriptomic technology called MERFISH to characterize the spatial organization of distinct glioblastoma subpopulations. In this case, again, we can do our UMAP, we can do our identify transcriptionally distinct cell types and GBM substates, but of course now we can visualize them also in the tissue and then perform various statistical analyses characterizing the spatial organization of, let's say, immune cells with respect to different GBM subtypes. So this is older work, but I think today what I really want to do is, teach you all a little bit about two methods that we have developed and how we are putting them together to discover something that was very unexpected. And perhaps through that, encourage you all to think about how you can apply these tools to your own data and questions of interest. Okay, so let's jump into it. So this journey started when we and others are now applying these technologies to generate, let's say, similar tissue spatial transcriptomics data sets, but across many different technologies or across many different health and disease axes. And one question that we come to is we want to begin making, let's say, comparisons regarding molecular and cell type composition at matched locations across these tissue samples. So again, in this case, I have two mouse brains, but if I really wanted to interrogate, let's say, the composition at this particular segment of the dentate gyrus in the hippocampus, and what's the analog here, or likewise a particular region in the thalamic area here versus here, it becomes a little bit challenging to make sure that we're comparing apples to apples. So this was a project led by my student Kaylin Clifton and Manju Anand in collaboration with Daniel Tord at UCLA, where we developed a tool called, we eventually called STalign, that builds on something called large deformation diffeomorphic metric mapping, which has been previously used in the MRI field to align structurally spatial omics data sets based on their shared features, such that we can then enable this type of matched spatial location. So I'll try to explain to you how this works. So consider we have two spatial omics data sets. Hopefully the lighting is sufficient for you to see. But generally, these spatial omics data sets, after they're segmented, quantified, and so forth, we can think of them as just points in space. So the first thing we need to do is actually, there's typically hundreds of thousands of points that we're trying to match across these two tissue sections. And the reality is there's often varying numbers of cells, across two different conditions just for biological reasons. So there's often not a perfect one-to-one matching. Therefore, rather than trying to align these 100,000 cells to 100,000 cells, which is a very computationally intensive problem, we decided to use a process called rasterization to reduce these data sets back into smooth image representations. And what we mean by this is we're essentially convolving a Dirac function with a Gaussian kernel to create this image representation of the spatial transcriptomics data set that is what we characterize as smooth. So that then allows us to essentially make the image representation typically has fewer pixels than the number of cells, so it is a computationally more tractable problem. But essentially, what this means is now I have two images, and I'm trying to solve for a function phi that when I apply it to the source image, it makes it look more like the target. So that's what this equation means. And this phi has a component that's an affine component and a diffeomorphic component. So how do we solve for phi? Well, that's where we use gradient descent, and we're essentially trying to solve for a particular function or try to minimize this particular error term, which, to break it down for you, it has a regularization component and a matching component. So again, these are two pictures. You can think of it as my two hands, and I'm trying to find some kind of affine transformation that will make them look like each other. So perhaps first I'll try to do a rotation, then maybe I'll do kind of local diffeomorphisms. But essentially, what the matching function is trying to do is quantify how similar these images are to each other. Yes, question.
Just a really basic question. So you're only working with the anatomical image at this point, not gene expression.
Excellent question. Yes, we are only dealing with the cell density at this stage. And there's a reason for that I will get into. The reason actually is because we typically want to make comparisons in the gene expression space. So if we incorporate that gene expression in the alignment itself is a bit of a double dipping problem. But we have explored actually, you can create this rasterization by incorporating the transcriptional information. You can imagine the amplitude of your Dirac function could be either a PC or the expression of some genes. But then you're incorporating that gene expression into your alignment itself, so that it might not be too kosher to evaluate the transcriptional similarity and difference. Great question. For now, we're just looking at cell density and trying to align these structures. And again, we're trying to solve for this phi such that when I apply it to this source, it makes it look more like the target. So in theory, this matching term should be going down, being minimized, but it needs to be subject to certain regularization constraints such that the overall transformation is smooth, which just means that, for example, if I'm aligning my hand, I don't chop off my thumb and move it over here and create discontinuity in space. And we believe this is a reasonable assumption when we're aligning tissue sections, because in general, we're not chopping things up into little pieces. So then what we learn in the end is, again, we solve for this phi using gradient descent, and what we end up learning is a diffeomorphism that is essentially, a field that we can then reapply onto our original XY coordinates and move those XY coordinates into their new aligned coordinate space. So now we get, we learn this field, we apply it to the points, we move them to this source aligned coordinate space. And of course, it's great to look at equations, but it's also just fun to look at the animation itself of this process happening. So just to see that again, I hope you can appreciate there are these linear affine transformations, but also a lot of local diffeomorphism necessary to especially get, let's say, the hippocampal region to align well. But once we've made this alignment, now we can do all the fun downstream things. We can look at the expression of different genes and begin quantifying their transcriptional similarity. And likewise, we don't have to limit ourselves to just within technologies. We can also look across technologies. And this is also where excuse me, our focus on structural similarity and imaging is quite important because let's see, so consider you have a single cell resolution spatial transcriptomic imaging technology like MERFISH and a spot-based resolution a technology like Visium. In order to align the two, you can, if you want to align cells to spots, it's a little challenging. So instead, what we focus on instead is aligning the image representation of the MERFISH with the associated histological image of the Visium, which is at a sufficient single cell resolution to be able to do this image alignment. So again, once we've done that alignment, now we can compare apples to apples. We have our aligned, we have our aligned merfish and also our Visium spots, and we can, for example, again, look at shared genes, which in this case is limited by the Visium or merfish, but we can appreciate, just visually for now, what are the, let's say, resolution and detection efficiency differences between these technologies. And of course, we don't have to limit ourselves to just the molecular information itself. We can perform single cell clustering on the merfish and identify our transcriptionally distinct cell types. And we can perform deconvolution analysis on the Visium spots, in this case, using our tool STdeconvolve, where now we've represented each spot as this pie chart, which visualizes the deconvolved proportions of each cell type. But I hope you can appreciate visually, we can now begin quantifying and comparing, let's say, at this spot, what is the proportion of a particular cell type? Is that consistent with what we see in the merfish? And vice versa. And of course, this STalign is relying on structural similarity. And therefore, since I think a lot of folks are interested in pancreatic cancer and prostate cancer here, we can apply these tools and technologies to a cancer setting. But of course, to do this type of structural alignment, it's only really makes sense perhaps in the context of serial sections in a cancer setting, because if you take it from different patients, they're not going to be structurally that similar anymore. But we're very excited actually to use serial sections and essentially take serial sections where perhaps one section we subject to spatial transcriptomics, one section we subject to spatial proteomics, one section we subject to spatial epigenomics now, and essentially use these technologies. They do offer readouts at different resolutions. So trying to do some type of higher order, what we call manifold alignment, which might require various assumptions about how gene expression relates to accessibility, relates to proteins. You can try to do this kind of dimensionality reduction and clustering in this higher order manifold space. But we're actually very excited to use physical space as the anchoring point. So when we subject these different tissue sections to these different technologies, can we use physical space to register them back together and begin integrating across these multi-omic platforms by using, again, physical space as the anchor. So if that's something of interest to you, definitely I encourage you to reach out, and we'd love to collaborate. Okay, so with that, just to summarize the first part, again, to make molecular and cell type compositional comparisons and match spatial locations, we've developed STalign to perform these structural alignments using diffeomorphic metric mapping. Certain details I didn't get into, we can accommodate partial matches. It does require structural and landmark similarity. We can also align 2D to 3D. So if we have light sheet imaging, we can register 2D sections back into our 3D. And I encourage you to check out our paper for more details. Okay, so it's part one, but now, now that we have these aligned tissue sections, how can we further, let's say, quantify their similarity or agreement, or identify discrepancies and differences that might be, indicative of some disease-specific process? So again, even though we've aligned them, there are, let's say, differing numbers of cells across these two tissue sections. So it's not always a one-to-one cell correspondence. So therefore, what we did originally on the paper and now sought to formalize more is we can create these grids, either hexagonal or square grids on the tissue, and essentially aggregate the gene expression within that grid. And this grid is matched across the target and the source. And we can do this kind of bidding in a cell type specific manner. But effectively, what that allows us to do is now we actually have these two aligned data sets. We have kind of gene expression at match locations. And we can say, at this particular pixel, versus this particular pixel, what is the gene expression? And through that process, we can, do across all the different pixels. There are some pixels that have high expression in this tissue section. It has high expression in that tissue section, represented as a scatter plot. And now we can calculate various statistics on the scatter plot representation that incorporates the magnitude similarity and the spatial information. So of course, in this case, we chose to quantify this as a cosine similarity. But what's useful is, of course, again, keep in mind, we're making measurements for thousands of genes. So we can summarize that across thousands of genes and look at their distribution and compare that for different pathways, different groups of genes, and see if there are some genes or groups of genes that exhibit more consistent similarity than others. So this is something we've sought to formalize a little more in a package called SE-raster, led by my student Gota Ihara, who was a postbac, now a PhD student at Caltech. But yeah, he developed an ARP package that's bioconductor compatible. And what this really is doing is, again, creating this grid on the spatial omics data set and doing this aggregation. And it turns out this kind of aggregation is very useful for not only comparing these, in this comparative context, but also simply for downsampling. So I think, again, for the single cell audience, some of you might be familiar with this challenge of as we scale these technologies to millions of cells, it becomes really hard to analyze everything. So what Bonnie Berger's lab did was develop geometric sketching as a way to downsample the data from the higher order manifold while keeping reasonable representation of all cell types and cell states. We can think of a similar idea here where, for example, let's say I want to identify spatially variable genes. Let's say I want to do that in a whole tissue context. So I can do my spatial variable gene expression analysis on the single cell resolution, or I can aggregate in these spatially contiguous hexagons and do that at 50 micron resolution, 100 micron, 200 micron, 400 micron, and so forth. But I can run SVG detection with this spatial SE-raster preprocessing. And in this case, as you might expect, when I aggregate, I'm essentially downsampling. I'm reducing the number of spatial positions I have to consider rather than looking at SVGs at a single cell resolution. I can plug this into NNSVG. I can plug this into our package Meringue. We can do CRAWDAD analysis on the pixel level and look for spatially variable genes across pixels. And because there are so much fewer pixels, that just drastically decreases our runtime and also decrease the memory necessary. But interestingly, for the most part, if you look at the ranks of the spatially variable genes we identify, on the x-axis is the single cell ranking of the spatially variable genes, y-axis is x-axis is the spatially variable gene rank at each resolution. For the most part, they're highly consistent, which means that the spatially variable genes that we identified at single cell resolution, they're still spatially variable with 50 micron aggregation, and it's much faster to run. And when we compare this to other downsampling approaches like geometric sketching, we see that if we had just downsampled using geometric sketching to the same number of pixels that we would have gotten through SE-raster preprocessing and aggregation, these downsampled data sets, when we look for SVGs using the downsample data, we generally see a drastic decrease in the true positive rates. But for this aggregation-based approach, we maintain a pretty good true positive rate, even for very coarse resolutions of aggregation. So we do think if you're looking to reduce the number of spatial points that you're considering, that this type of aggregation preprocessing can be very useful. And one limitation, though, is that it does lead to a decrease in the true negative rate. So you get some false positives. The way that a lot of these true or false positives are driven by edge effects. So for example, the particular way in which you partition your cells into these grids can create little edge effects. But it turns out we can get around that by doing permutations, which we do by way of rotating the sample. So when you rotate the sample, the grid changes a little bit. So then your aggregation is a little bit different. And it turns out that's enough to split up, let's say, cells sometimes that contribute to these false positive patterns. So if we restrict to, let's say, only spatially variable genes that are consistently identified as significantly spatially variable across all permutations, now we get a very good both true positive and a true negative rate. So that is an approach we can use. Yeah, but the reason we sought to pursue this was initially when we and others were trying to apply a lot of these existing tools to these newer data sets. They just kind of poop out. We have millions of cells now. For those who are doing Visium HD, this is also true. Sometimes it's just very hard to run on your Mac laptop computer. You would need to have access to a high performance computing resource, which not everyone might have, or maybe you just want to do a quick preliminary thing for your grant. So with SC-raster preprocessing of these aggregation, now we can identify spatially variable genes in a matter of minutes. And in this case, appreciate how there are spatially variable genes that occur at a tissue level, that a lot of it is driven by, let's say, just organ-specific organization. But then we can also do rasterization on each organ independently and look for spatially variable genes, let's say, within the kidney. Okay, great. Other fun things that are enabled through this type of aggregation, we don't have to aggregate just genes. We can also first perform single cell resolution clustering analysis, identify our cell types, but then ask if we bin these, create these little grids and bin these cells into those grids, are there certain cell types that tend to be binned together more than what we expect by chance? So it turns out that that's a type of analysis that previously has been done a lot in ecological research. So we were actually able to then integrate our SC-raster preprocessing with this co-affinity analysis, co-occurrence affinity analysis that somebody else developed to look for whether trees and deer, co-localize together. But in this case, we're looking at whether one cell type is more likely to be found in the same pixel as another cell type, and to do that for, of course, all possible cell type pairs. And in the case of this embryonic mouse, when we do that, perhaps unsurprisingly, in this case, what I'm visualizing is for each transcriptionally distinct cluster of cells that we identify, if we evaluate their co-occurrence, red is high co-occurrence, blue is low co-occurrence, you can see it forms these little triangles. I guess they look kind of like TADs for those who like TADs. But essentially, it's representing similar things where these cell types are more likely to be spatially observed together. And if you visualize where they are, perhaps unsurprisingly, they correspond to different organs. So we're very nicely able to recapitulate that, the different cell types co-localize together because they're functionally distinct in different organs. So these are just a few of the different analyses that we are able to enable with this type of aggregation-based preprocessing. And now, what we're very excited about, again, is actually applying these tools like STalign and SC-raster to clinical and disease contexts. So we're very fortunate to be collaborating with Hamid Rab, who's a nephrologist at Hopkins, where we've been able to do spatial transcriptomics on a murine model that he has, where we can have healthy control mice, but also mice that have been subjected to this reperfusion injury that is a model for acute kidney injury. And we can, again, these are two Visium data sets of sagittal sections of the mouse kidney And we can align them, we can aggregate them into bins and look for genes that have, let's say, high cosine similarity. And in this case, this is one example where visually, I hope you can appreciate, this gene is highly expressed in the cortex and not so much in the medulla. And it's very consistent across healthy and ischemia. So we get a very nice positive correlation and cosine similarity. But because we have these tools, now we can screen the whole transcriptome and evaluate all the genes and specifically look for genes that might change in their spatial patterning. And in this case, there are definitely genes that are not changing, let's say, globally in their magnitude. So if you just did bulk RNA sequencing, this gene would be very comparable and the controls versus the ischemic. But by looking at it in a spatially aware manner, we can appreciate that, there's a real change in that, the spatial patterning of this gene. So this is something we're continuing to work on. So hopefully I'll be able to share more in the future. So as a summary of part 2, again, this aggregation preprocessing enables comparisons across aligned tissues, but we can also use it to reduce the number of spatial locations to enhance runtime and reduce memory. We do like doing this generally in a cell type specific manner. So we do our single cell clustering first, and then looking at only a particular cell type, we can aggregate and do this type of evaluation to take advantage of the single cell resolution component. But we can also look at not only aggregating gene expression like continuous variables, but also categorical variables like cell types. So we've implemented this in an R package called SC raster, and it's available on Bioconductor now with the latest version of Bioconductor. We encourage you to check it out for more details. So this is everything you've learned so far is all just background to take us to this final story, which we just shared a few weeks ago on bioRxiv. But what we really wanted to do when we sought to pursue this project was, and this is work led by my student Caleb Hallahan, who's just passed his DBO last Friday. But what we really wanted to do is actually characterize the advantages and disadvantages of these technologies. So there's so many of these spatial omics technologies. Let's see if we can apply the tools that we've developed to actually do a real quantitative comparison. So fortunately for us, there was this previous paper from 10x where they took serial sections of a breast cancer tumor. So these technologies are destructive. So that means that if you take one cell or section for one technology, you can't use exactly the same tissue section for another technology. But if you do serial sections, that's probably as close as we can get to a perfect replicate. So what they did was take these serial sections and subject one of them to Xenium, which is their imaging-based transcriptomic platform, which again is targeted. It's limited to a couple hundred genes, but it is single molecule, single cell resolution. And they subjected another one to their Visium technology, which is capture and sequencing based. So it's spot based, but sequencing, so you get full transcriptome. So we just wanted to compare and see if maybe there's some way to integrate across these technologies, yada yada. But so what we were again able to do is use our tool STalign to align these tissue sections. So they've already been aligned, which is why they look so similar to each other. But of course, now we can precisely identify the region that's shared. So even though they're serial sections, slightly different parts of the tissue got characterized by the various spatial omics technologies. So presumably, again, these are different technologies, but they're probing for at least some of the same gene features. So for example, if we look at, yes.
This slide.
Yes.
This is, you can even do FFP versus frozen as well with these two tools.
Great point. Both of these are FFP samples. So this is, great. Excellent question. So this is the Visium site assist FFP based. So we're not doing polyadenylated sequencing anymore. Great. Great. So again, yeah, we can look at the same genes, for example. Yeah, this gene happens to be profiled by both of these technologies, and we can visualize how they look in the tissue. Again, Xenium single cell resolution, Visium spot resolution. It's a little bit visually hard to see the similarities and differences, but it turns out, if we aggregate to the same resolution, now hopefully it's visually more apparent to you all that this region of the tissue has high MS4A1 expression by Xenium, and likewise also has a high MS4A1 expression by Visium. And for the most part, we see pretty good spatial correspondence. And yeah, these are B cells. So presumably B cells are organized in these regions of the tissue. And we can further quantify that, again, using the approaches we've developed before. Yes?
Using the rasterize tool, can you define the resolution that you're rasterizing to?
Great question. This is 200 microns.
Within the software, it's possible.
Yes, that is a hyperparameter. Yeah, that we can pass into the tool. It's actually a very interesting line of area of exploration to characterize different spatial patterns at different scales. So we are using that tool to see what happens at a 50 micron resolution versus 200 versus 400, so forth. Yeah.
The gaps in the visibility.
Great question. In this case, we aggregated, actually wonder if he did consider it. We know exactly where the Visium spots are, and we're assuming a 55 micron round circle. So then we can aggregate the Xenium to the same circles to enable a fair comparison.
There are empty gaps between.
Yes.
Point. Do you assume expression of the empty gap, or do you just aggregate all of the sampling spots?
Great question. Yeah, if we're aggregating to a really coarse resolution, yeah, we would be missing things in the empty spots. Yes. So that is in theory, maybe if there's more expression in those empty regions that are not captured by the Visium, we should see slightly more detection in the Xenium for the matched locations. Great points. But yeah, for the most part, they do look quite similar. However, there are other genes like TUB2B that really piqued our curiosity because in Visium, it looks like this. It's a little hard to see what happens in the Xenium, but if we aggregate to the same resolution, now hopefully it's visually a little more clear. Unlike MS4A1, here they have very different spatial patterns, let's say. We get very high expression in this region of the tissue that we don't see any here. So this again, piqued our curiosity. So what is the difference between, what is explaining the difference between these two technologies? We're using them to profile roughly the same tissue, but they're giving us very different results. So in order to understand, the potential source for these discrepancies, we do have to understand how these technologies work. Just as a brief explanation, the way Xenium works is that they use padlock probes that are complementary. This is my doodle of a padlock probe. So these are their probes. And in theory, they can form these padlocks that have these arms that are complementary to a target RNA of interest. So then when ligation happens, you can get rolling circle amplification, and the rolling circle amplification has a sequence that has a readout probe that can hybridize. The readout probe has a fluorescent tag. So these fluorescently labeled decoder probes can hybridize to the RCA product. And then that's what allows us to take a picture and we can see a really bright spot. And through many rounds of imaging, we can decode the optical signature that corresponds to a particular target gene. And that's how we're able to achieve high degrees of multiplexing to be able to detect hundreds of genes simultaneously. It's worth keeping in mind, therefore, the accuracy of the gene expression profiles that we're characterizing by these probe-based approaches really relies on the specificity of our probes to bind their intended target gene. And yeah, so that's where it becomes important to understand what is off-target binding. So we define off-target binding as just when the probe binds to something other than the target gene, which can be a different gene. So for example, if these probes hybridize to a different gene than our intended target gene, once the ligation and the rolling circle amplification happens, the final fluorescent product, we really can't distinguish it from the original, from the true on target. So this is all just to say, off-target binding can ultimately distort the quantification of the target gene's expression, because in this case, even though there's only one copy of the target gene, because the probes are binding to this off-target gene, I would count it as two. So it's going to distort the target gene's expression quantification, because the observed expression is really a representation of both the on-target as well as the off-target gene expression. So, okay, so how do probes know what to bind? This is just some background for students in the audience who might be less familiar with the biochemistry. So remember, we have this wonderful complementarity system in our DNA where A pairs T and C pairs G. So when we design these oligonucleotide probes, they have a particular sequence that we design to be complementary to the target RNA of interest. So therefore, if we wanted to look for off-target binding, this essentially becomes an alignment problem. So we can, so this is where we collaborated with Hayden G. and Steven Salzberg's lab to develop this tool called Off-Target Probe Tracker, which essentially is just an aligner that references, compares against annotated references to predict off-target binding. So how it works is, again, we can take our probe sequences. In this case, we just took the 10x probe sequences. These are, again, commercial panels. So we can take the probe sequences and we can align them to the human genome and see where do these probes putatively align based on base pair matching. And then we can reference various human genome annotations like RefSeq, GenCode, and see how, which genes do these aligned regions fall into. So for example, when we look at the probes for TUB2B, here's an example of a probe. It indeed does align to TUB2B. So this is the exon of TUB2B. This is where our probe aligns. These are the introns. But interestingly, it also aligns to TUB2A. So this is, again, where our sequence is. And there is a region in the exon of TUB2A that this probe does align to. So so presumably, therefore, we speculated that this probe could also bind this region of TUB2A. And actually, when we look at the expression, so again, this is reminding you the expression of TUB2B. There's not a lot of correspondence. But when we look at the expression of TUB2A, now things are looking much more similar, right? And we can even aggregate the signal from TUB2B and TUB2A together. And I hope that it's visually apparent that at least the spatial pattern of the aggregated expression of TUB2B and TUB2A in the Visium is visually more similar to the spatial pattern of TUB2B in the Xenium, which is consistent with our prediction that the Xenium TUB2B probes are exhibiting off-target binding with TUB2A. So of course, now that we have a tool, we can apply this to all the different genes in this particular gene panel and compare them with, in this case, the total count expression in Visium versus Xenium. And for the most part, I guess we were trying to understand how this could have been missed before. So it is worth noting that on a total gene expression level, when we consider only the overlapping regions between these two tissue sections, we do observe a very nice correlation. And further, we don't really observe an obvious trend with the gene expression magnitude and the presence of off-target binding. So for example, it's not like the probes with off-target binding are consistently way higher. in the xenium than the visium. So it's definitely not obvious. And this is in part because a lot of times the off-target genes are not really expressed in the tissue. But yeah, we kind of speculate that such off-target binding might have been previously overlooked due to this high correlation. So yeah, this is just a word of caution that perhaps we note that correlation between technologies is a sufficient metric for consistency, but or it's a necessary but not sufficient metric for consistency, and that we should scrutinize individual data points. So this is the published figure where they note that this technology works well because there's a high correlation. And again, we're able to recapitulate it quite well. So you can see our point, their point, their point, our point. So this is supposed to be a one-to-one recapitulation. But you might notice there's a particular point that's missing from their figure. So actually, there's this gene ApoBec ApoBec3B that is not detected at all in Visium, and it is detected quite highly in xenium. And again, if we visualize, we can again do our off-target probe binding predictions, and in this case, again, visualize their patterns in space, and we can aggregate the expression of all the off-target predictions, and for the most part, we can see a comparatively more similar spatial pattern once we consider the off-targets. So again, we do think this gene is also impacted by off-targets. And there's many other examples that we note in our paper. For example, CCAM8, again, very disparate pattern. Once we aggregate the off-targets, a much more similar spatial pattern. So in that sense, so this is the final component that I hope I've been able to show you how our comparative analysis is able to prevent evidence of off-target probe binding affecting the 10x xenium V1 human breast cancer gene expression panel. This is a panel that's commercially available. And even though here we did focus on 10x because of the availability of their probe sequences, there are other companies that don't make their probe sequences available, so we weren't able to interrogate this. But also, there were these orthogonal data sets that we were able to integrate to substantiate our predictions. So we don't exclude the possibility that this off-target probe binding might affect other probe-based detection approaches. And finally, we provide this OPT software as a tool that we encourage you to try out and apply to your own data sets and technologies of interest. So just some recommendations going forward, I think, for the technology development folks in the audience, if you're designing probes, we do encourage you to use tools like our off-target probe tracker to test for these off-targets. One thing that we emphasize in the paper that I didn't really emphasize in the talk is the importance of using many reference annotations. So these annotations do change over time. So sometimes there's a probe that is, what is considered a gene and an exon versus not. Different references can have slightly different boundaries on the exons, which evolve over time. It is an active field in itself. So we do encourage you to try out multiple annotations. But if you already have data generated using these probes, which we do, I think for us, if you're training foundation models, as we are trying to do also, I do encourage you to omit these genes to avoid error propagation. I'll just leave it at that. If you're performing integrative analysis, so we were actually trying to integrate this data with single-cell RNA-seq data. The way that we're going about doing this is rather than integrating with TUB2B and xenium and TUB2B and single-cell, we're actually integrating TUB2B and xenium and TUB2B plus TUB2A in the single cell to incorporate these off-target predictions in the manifold alignment itself. And then finally, of course, we're interpreting these results. Just know that these, if you see differential expression in TUB2B, maybe it's driven by TUB2A, and to incorporate that into your interpretation. And in general, in our preprint, we have a lot more recommendations for probe redundancy and other design considerations. Okay, So lastly, I'll leave you with some food for thought. Again, these are commercial platforms. They've been available for some time. As of December 2024, over 16,000 of these Xenium consumables have been sold. Each tissue slide profiled costs about $5,000, which I think underscores this platform's widespread use, but also its high commercial value to 10x. 10x Genomics, which is the company that makes Xenium, is a publicly traded company with a market cap of over $1 billion as of March 2025. And evidence of this off-target probe binding was not previously identified despite a publication from 10x researchers comparing across these technologies. So I hope this last story exemplifies how the, let's say, complementarity between industry and academia fosters a more rigorous, transparent, and reliable scientific process, ultimately to the benefit of all of us consumers and even the public. And in this moment when federal funding for academia for science is being threatened, I do hope this last story highlights the critical role of academic researchers in ensuring the robustness of these industry-led product development by providing oversight free of financial conflicts of interest through independent federal funding. So with that, I would just like to thank my independent federal funding and all of you for your attention, and yeah, happy to take any questions. Yes, please.
First, I think even with the multiplex coefficient data or massive monotonic element in situ database, still find challenging when compare gene expression in study the distribution and probably designing probe, right? Like what tissue express something. And I feel like the first part you mentioned, STalign or the SE raster, has a great potential to integrate those massive monotonic experiment into one, like integrated atlas for the transcriptomics with thousands, hundreds of genes. It can be a supporting evidence for the provision approach, although they only screen for 1,000 genes as a panel. Are you working on that, or is that some direction you will try?
Yeah, great question. Just to summarize for the audience and those, I don't know if the folks on Zoom can hear the question, so I'll try to summarize. So let me know if I've misunderstood. So your question is like, Allen Brain Atlas and these other large consortia are generating these gigantic Murfish data sets for thousands of genes, and do we anticipate that tools like STalign and SE raster can help kind of integrate them into one common coordinate reference that in the future we would be able to align, let's say, new data sets into? Yes, I do think that that is a potential application. And that actually is the reason why we make our tools kind of open source and available. It's not something I'm personally planning to do because, yeah, for us, we're much more interested in disease comparisons. And yeah, so I guess I'll leave it at that.
Thanks.
Yeah. Yes.
The chart, think about the Xenium versus the recent chart. You did the comparison using the average count, right? So to say some genes are entirely this way. Would that be more drastic if you use it more like density based, considering how each pixel is represented differently? Because I thought, for example, the top B1, you totally convinced me that one is totally different. We'll be probably popping up into a different core.
Yeah, excellent question. Yeah, so just to summarize for the folks on Zoom, yeah, this I'm only looking at, for the shared region across the two tissues, what is the total expression of each gene? And if we had done a more spatial pixel-based cosine similarity or correlation, TUB2B should jut out a lot more because that cosine similarity should be much lower. And I guess we did do that. We did take a look. The reason for this particular visualization actually is simply to highlight the consistency with the publication and the inconsistency. We have tried, looking at the correlation or cosine similarity in space. It turns out to, there's a couple of challenges with that metric. Correlation in particular and cosine similarity is very impacted by magnitude. So there's, so you have higher cosine similarity or higher correlation just because the gene is more highly expressed. So there are some lowly expressed genes that have really obvious pattern differences that are not being captured very well by that particular metric. So that is an area we're actually continuing to develop in.
Sorry, if I may follow up, there's actually another important question in my mind. I think that SE raster is really cool. But how do you normalize signals coming from different technologies? Like, because there are zeros and like high expression will be looking very different. I know some of the chart, like they're all within like 1 or 2 levels of difference, but like, do you actually account for dropout rate or like detection sensitivity?
Yeah, it's a great question. Yeah, how do we compare across technologies considering, there might be different normalization differences. When we compare across technologies, like in our STalign paper, we actually did normalize. So yeah, we can do count CPM type normalization for the sequencing based. We can do volume normalization for the imaging based, and just make sure that they are not confounded by different unique artifacts that are in each technology. Great question. Yes.
Hey, very nice talk, and thanks for, I guess, bringing about some important what should be housekeeping things should do while we are using some of these wonderful technologies. I have 2 questions. One is more technology associated, where in the STalign, it's, as an imaging person, sounds a lot like a registration problem.
Yes, it is.
What is, or is there an extent of similarity limit to which STalign can work?
Yes.
That is it going to break down, that can it work?
Great, excellent question. Just to reiterate for the Zoom folks, is there a limit of the, in some ways, like registration accuracy for STalign? And the accuracy is limited by the width of our Gaussian kernel that we use to rasterize. So if we are creating this image representation at 30 micron resolution, that means if you have a structure that is smaller than 30 microns, it becomes a blur. So for example, consider my hand. If I'm creating an image-based resolution that's kind of like blurry, if I align, I'll probably, for the most part, align like whether it's on my hand or off my hand, but my fingers might not be perfectly aligned. So there is so far I think we've been, I would say, very successful at aligning if it's more than 2 cells wide. If it's a really thin structure that's 1 cell wide, that is very challenging for us. Great question.
That's great. I have a follow-up question that's more on the biology, especially in your IRI kidney injury model.
Yeah.
And there that you had a very nice finding about CRYAB, right?
Yeah.
Which its spatial pattern looked very distinct across your control and ischemic kidneys. Do you know why, or what could it be?
Yes. So this is ongoing work, but our speculation right now is that yes, there's local injury responses, but there's also why would these shifts happen? We see lots of patterns that are like, oh, it's expressed in the healthy and it's not expressed in the control, or it's not expressed in injury because that either the cells die or the cells are turning on inflammatory response programs, not really able to do its healthy function, and also vice versa. There are also lots of genes that turn on in injury and you normally don't see in health. The ones that have shifts, a lot of them are metabolism related. So we think it has to do with the local metabolic demand. So the medulla is typically in a more ischemic environment anyway, whereas under injury, now the cortex is also more in an ischemic environment. So there are these various metabolic shifts that we think are happening, just because of the newfound ischemia.
But I
Yeah.
think there might be a link to protein homeostasis as well. Just want to throw it out.
Yeah, interesting. Yeah, I think that's where it's really fun to do this, big data search, so we can actually look for all the genes that are changing in their spatial pattern, run our gene set enrichments, and do that in compartment-specific manners. And definitely a lot of fun things to dig into. But yeah, if you have hypotheses, I'd love to look into it. Great. Yes.
About STalign. Does it have output that tells you how good of a match the realignment is, and can you use it for like a best match kind of problem? Like, could you pair or realign an image with 4 different other images and say, okay, well, this is one that's the best fit?
Oh, that's an interesting problem. Yeah, the question for the Zoom folks are, is there a way to tell if the alignment is good? And if you have many different possible alignments, quantify which one is better. In theory, yes. So we are doing a gradient descent and quantifying an error term. So I would expect, let's see, like the best alignment should probably minimize this. Just have the highest matching, and I guess just have the lowest energy or error term. However, that is also subject to this mixture model component where we try to fit what is on the image versus the background. So I can imagine if there are some tissues where, let's say your alignment is actually quite good, but maybe one tissue has a bit of a tear, so then the matching component is slightly not comparable, then that will lead to a slightly different final error quantification. But the way, I guess to answer the first part of your question, how do you know if we are doing okay? The way we've tried to evaluate is based on the convergence. The gradient descent kind of plateaus. So we're kind of at an asymptote where we're not really doing any better. That being said, though, I guess this is maybe if folks are interested in contributing to the package. I think right now we use a very, let's say, not smart gradient descent where we're just taking equal size steps. So therefore, we can get stuck in local minima if people are familiar with gradient descent. So I think if I had a motivated student, maybe we would do more dynamic step sizes and try to find global optima as opposed to a local optima.
When the STalign output, is it easy to access things like the error term for the?
That's a good question. We don't output it per se. Maybe we should add a verbose parameter to output it. It is implemented in Python, and hopefully the code is commented well enough that if people have made modifications on it to actually output what they need. I think we didn't output as many things as we probably should have. For diagnostic purposes. So definitely encourage you to take a look at the code and feel free to make a pull request. We'd love to have you as a contributor. Great. Awesome. Thank you all.